from Professor Thompson's group. My research is about pH-sensitive photoluminescence of quantum dots in human ovarian cancer cells. Fluorescent probes are widely used in biomedical studies. They can label a specific component in biological sample. From this image, we can see cell nucleus was stained with blue color and actin was stained with purple color. So fluorescent probe can help visualization under normal optical microscope. There are two main types of fluorescent probes. One is organic fluorescent dye and the other is semiconductor quantum dots. The organic fluorescent dye is notorious for photobleaching. We can see from this image, the cell nucleus stained with green color will totally change within 3 minutes. However, the quantum dots show as the red color can keep fluorescing within 3 minutes. So quantum dots is photostable and they become a promising material in biomedical studies. In this talk, I will first give a brief introduction to quantum dots, and then I'll mention how we synthesize the quantum dots, which are biocompatible. And then we deliver the quantum dots to mammalian cells by endocytosis. And finally, we study the quantum dots pH sensitivity. Quantum dots are semiconductor particles, which has a size at few nanometer range. And when we use UV excitation to excite quantum dots, electron hole pairs can be created and confined in this small region. Due to the quantum confinement and the small size, the electron hole pair can be recombined and emit visible light. To achieve emission in visible light range, the common use material for quantum dots core is cadmium selenide, cadmium sulfide, and cadmium telluride. And outside the core, we can add a shell, which has a wider bandgap to have a better confinement. The material used for shell is zinc sulfide and silica. And we can also add a cap molecule, which is a hydrophilic molecule that can make quantum dots water soluble to apply into the biomedical studies. In order to have total control over the property of quantum dots, we synthesize quantum dots right here in post-power group. The quantum dots have unique optical properties, which is different from organic fluorescent dye. From this figure, we can see the fluorescent dye has a narrow absorption. So we have to use one specific light source to excite the fluorescent dye. And its emission has a long red tail. So it is difficult to use many different color fluorescent dye at the same time. However, the quantum dots has a broad absorption and relatively narrow and symmetric emission. So we can just use one excitation source to have multicolored emission. Besides that, quantum dots is stable, non-photobleach. And we can see this picture is a quantum dots that I made. The quantum dots have size-tunable emission. For the smaller size, emit green color, and for the large size, emit red color. The quantum dots that we made is cadmium selenide as the core, and I use a double shell. One is zinc sulfide, and the other is zinc selenide, and the other is zinc sulfide. The double shell structure can have a better optical property than a single shell structure. And the produced quantum dots that is only dissolved in organic solution, such as a chloroform or toluene. After we change the copy molecule to macatoacetate acid, MAA, and we can make quantum dots water soluble. Because of the copy molecule is macatoacetate acid, so I call this quantum dots MAA quantum dots. dots. I also characterize the quantum dot size by using TEM, and the size is around 3.6 nanometer. In the experiment I'm going to talk about today, I use these MAA quantum dots. The MAA quantum dots can be simply put into the cells by endocytosis without any help of biofunctional ligands or any permeabilization steps. So we just need to mix our quantum dots with the cells, and we keep mixing for 24 hours. And from this image, we can see the quantum dots are internalized with the cells. And we also washed away the quantum dots which is outside the cells. So from this image, all the red color comes from quantum dots inside the cells. And this image, we can have a close look that the cells can take a lot of quantum dots. And we keep absorbing these cells for at least 2 weeks. And the cells' morphology appear normal. So which means this quantum dots is biocompatible, non-toxic to the cells. It turns out the photoluminescence of MAA quantum dots depend on the pH in aqueous solution. From this figure, we can see the initial intensity increases with increasing pH, and its behavior is almost linear, monotonically increased with increasing pH. We also want to see these pH-sensitive quantum dots in the cells. So we first mix the quantum dots with the cells, and because it is difficult to raise the living cell to a certain pH value, so we add the fixing agent such as formalin or formaldehyde to the living cell, and that can preserve the cells intact, but the cells are dead. So we immerse these fixed cells in the pH 7 buffer, and we can see the red color comes from the quantum dots inside the cells. And then we change the pH 7 buffer to pH 4. For 3 minutes, we can see the quantum dots decrease its fluorescence. And wait for 3 more minutes, we can see the intensity keep decreasing. And then we change the buffer to pH 7 again. We can see the fluorescence intensity can be partially recovered. And we also want to study the MAA quantum dots pH sensitivity in living cell. So we load the quantum dots with living cell, and we also use a traditional pH indicator, fix it. We load quantum dots and fix it together. And we use a drug, chloroquine. This drug can increase pH of intracellular vesicles. And after 30 minutes chloroquine treatment, we can see both our quantum dots and fixit emit brighter color, emit brighter fluorescence, showing that increase the pH in cellular vesicles. And besides that, our quantum dot is photostable, non-photobleaching. So quantum dots can be a potential intracellular pH sensor. A possible mechanism for MAA quantum dots pH dependence is the MAA quantum dots is negative charge, and the charge may be different under different pH buffer due to the charge balance between the quantum dots and the buffer. And at lower pH, some MAA may dissociate from the quantum dot surface, which can expose quantum dots to the solution and increase the number of non-radiative recombination pathways, and thus decrease the fluorescence. And without this MAA protection, quantum dots are insoluble and tend to aggregate. In order to verify our hypothesis, we ran gel electrophoresis. Gel electrophoresis is a technique that can separate particles according to the size and the charge by forcing them through a porous gel in an electric field. So we load our quantum dots in the negative electron part, and after we apply the voltage, the quantum dots will migrate toward the positive electron part. The migration mobility is proportional to the charge and inverse proportional to the size. And we use a DNA marker as a reference to check how far the quantum dots can migrate. And from this figure, we can see the quantum dots in basic condition. They can migrate faster than in neutral and in acid condition. And we can also see in acid condition, some quantum dots stay in the original position, which means the typing molecule MA dissociate from the quantum dot surface and make the quantum dots insoluble. And they change the aggregation, make the quantum dots stay in the original position and they cannot move. So this experiment supposes that our hypothesis, the charge on the MA quantum dots may affect the photoluminescence intensity, and the typing molecule MA dissociate from the quantum dot surface may in turn decrease the fluorescence. So in summary, we in-house synthesize the quantum dots which are biocompatible, and this quantum dot fluorescence is pH dependent in both aqueous solution and in fixed cells. And its behavior is monotonically increased with increasing pH. And we also observe the pH-dependent photoluminescence of quantum dots in living cells. And its behavior is similar as the traditional pH indicator. But quantum dots are not sensitive to photobleaching, so demonstrate our quantum dots has potential to be used as an intracellular pH sensor. This work is funded by Air Force Office of Scientific Research and the National Institute of Health. I would like to thank my colleagues in USC Post-Power Group and Professor Donaldson to make this work complete.
Angela, real good job. Martin, you look really handsome. Any questions? Any questions? Go ahead.
I have a question. You said is the quantum dot endocytosis in the cell. Have you done besides imaging any other experiment to confirm that it is endocytosis and which pathway of endocytosis, like which pathways they go through the cell? Is it platinum dependent? Is it caveolin dependent? Which kind of endocytosis is responsible for these quantum dots uptake into the cell?
Do you mean the endocytosis?
I mean, how sure you are they are endocytosis? Besides that, you had the image that they were trapped in vesicles. Do you have any other claim that they really endocytose into the cells, or is not another mechanism of uptake?
Okay. We mix quantum dots at 37 degrees C and 4 degrees Celsius. And because endocytosis is kind of energy driven, and we cannot absorb any endocytosis in 4 degrees C sample, but we can absorb the endocytosis and the quantum dots uptake by the cells in 37 degrees C sample. So this is a general metabolism, endocytosis.
Any more questions? Go ahead. Yeah. Go ahead.
Right here. You've been probably living quantum dot world for a couple of years, and you're probably totally immersed in the application of these things. It seems that what you've made is 10 to 100 angstrom, very small spheres coated with silica. Is that correct? You could put a silica SiO2 core exterior on these things?
Yeah.
Okay.
Do you mean I can put that?
No, you make these little spheres, and then you kind of a SiO2 boundary or layer or skin on the surface of them. Is that correct?
Uh-huh.
What other applications have you seen of these little tiny 10 to 100 angstrom spheres in the workplace or in the industrial world? Can you think of anything else beyond the application that you're currently working with them on?
For the silica shell, I don't use that, but according to literature, they are more biocompatible. But silica is in the red band gap, so the fluorescence intensity is not so bright.
What's the thermal stability of these little spheres that are being used? You talked about pH variations, etc. But if you start heating these things up, you get to about, say, 50, 60 C, what happens to their physical integrity? Does it collapse? Does it? Do you know anything about that? Or?
I don't understand.
If you take the small batch of spheres you've made and you put them in a test tube or something, and you start heating them up, you raise the temperature, what happens to them?
The fluorescence?
No, just in general, physically, what happens to them? Do they maintain their shape?
If we incubate the compound in 4 degrees C and increase the temperature to 37, we just mix for 1 month because cell cannot stay a long time in low temperature. So we have usually it stay in 37 degrees C.
All right.
So I believe the low temperature is not good for cells.
Good, thank you. Appreciate it.
Go ahead.
I think early on you showed a graph of photoluminescence versus pH, and the higher pH is up to 10, where the most photoluminescent. You described how the low pH can hurt the dots, but how can it improve the photoluminescence by making the solution basic? What changes to make it better if you put it in a high pH?
In high pH, because our quantum dot is negative charge, and in basic condition is also a kind of negative charge, a reach of negative charge. So that can make quantum dots, they are not aggregation. They disperse very well. So, which means the capping molecule, the MAA, protect quantum dots very well. So that keep the fluorescence.
So even at pH 7, you have a certain fraction of the dots are agglomerated and not contributing to photoluminescence.
In pH 7 is a normal fluorescence, but in higher pH they can intensify the fluorescence. So I guess it's because the negative charge can disperse the quantum dots better than in the low pH, pH 7.
Any more questions? Let's thank God all for it. What I will do now is